My lab is very interested in building and applying technologies on the sort of the area of functional genomics, that we want to understand how these genetic and genomic variants that we heard about earlier from this session translate to brain function as well as dysfunction in the context or in the brain in vivo, in an intact organism. And this is really inspired by the community of human genetics that put together these really incredible advancements in the past decade. Now we know the devastation of these devastating diseases and human brain disorders actually have very strong genetic roots. So from the one hand, we know that in the past decade, there are many high confident de novo variants. And these are de novo variants that's present in the affected children, but not in the family members, that has always been almost causally associated with neuropsychiatric disorders such as autism, intellectual disability. So this almost started over a decade ago, that multiple groups come up with this idea of de novo whole exome sequencing and identify recurrent and collective, rather short list of genes that actually contribute to autism and intellectual disability. And this list of genes has been reproduced across cohort, across different statistical framework, that in the last 2 to 4 years, you can start seeing the convergence of these risk genes. And in the last version of this list, there are 72 genes that their loss of function is causally associated with complex disorders such as autism. I want to emphasize that in the realm of human genetics, this is really the kind of genome-wide significance that we never had in the past. But understanding the long list of genes that their change or variation contribute to a disorder does not mean that we understand this disorder at all, because we do know that for many of these genes, they express in different cell type. They express at different time of this dynamical process of neurogenesis and brain maturation. And now we also know that these variation or changes in the genome can actually impact many different cell types. One gene can impact one or several cell type, and collectively, we really run into this many-to-many problem, that we have many risk genes, 72 or more, that each of the cell type can collectively affect one or several different cell types in our brain in the context of developing progress. So this is really the goal and the central thesis of my lab, that how do we understand the brain? Is autism or intellectual disability that comprised of these different destruction in different parts of the genome simply a neurological convergence? Or are there any ways for us to classify these different genetic variants? And by convergence, what I meant is that is it possible that some subsets of these 72 risk genes, for example, actually impact the same molecular pathway, or the same molecular pathway within the same cell type, or even within a very narrow time window of the development. And these are the information that is necessary to know before we start thinking about treatment or even ways to classify them together to develop therapy. So these type of specific information in terms of molecular pathway, cell type specificity, or also time window that they manifest will actually help us to use them as an entry point to understand the complex process of human brain development and learn how the brain works. So this started a few years ago when I was a junior fellow at Harvard and working with Feng Jiang, Paula Lata, and Aviv Regev, that together we came up with a way that hopefully is a systematic way or a sort of scalable approaches to use functional genomics and understand how long lists of genes contributing to brain development in vivo. So the strategy works as the following. We can use CRISPR that we learned earlier from Lacrosse talk, and we can deliver these guide RNA targeting your gene of interest. We need to do so using viral delivery into an embryonic, intact developing brain in vivo. And we can do so in pool, and days and weeks or even months after the in vivo intact development, we can harvest the tissue and look for these perturbed cells and use high resolution, high content, such as single cell RNA-seq method to profile for each individual cell and each individual perturbation. What are the cell type specific perturbation effects for each of these risk genes that we're interested in targeting? So the first version of this as a prototype was published a few years ago, where we actually apply and build this technology using the way of lentivirus to be able to analyze about 3 dozens of different risk genes that contribute to autism. And we have identified recurrent cell type and molecular pathway that's enriched even across this rather small subsets of risk gene, which is interesting. And at that time, we already started realizing that this seems like a really great approach. Why are not people using it? Why are people not doing more in vivo screening? And it turns out this is really a very sort of common challenges for doing in vivo experiments in general. It's just the limited numbers of cells that we can reliably label and then harvest later on for analysis from in vivo. And this is largely limited by the viral delivery, as I said. Many of us are using lentivirus for genomic work, which is wonderful, but they have rather limited permeability in vivo and then lower thermal stability to be used in an intact animal. So a few years ago when I started lab, and my first graduate student, Xin and I thought very deeply about whether there's any way to use an alternative viral vector. And one of the obvious choice that many of you may have used before, which is switching to adeno-associated viruses, AAV. And they are fantastic in vivo gene therapy approaches and vectors with minimal immune pathogenicity and can penetrate well, even for systemic deliveries to the brain, but also many other organs. So AAV is fantastic, but the issue really is usually you inject the AAV, you do a surgery, and you usually wait for weeks or even months because it takes quite a long time for AAV to express. Whereas I just told you that we're very interested in the process of corticogenesis, which the entirety takes about less than a week. So if we do the surgery and the virus doesn't take, it will take weeks to turn on, we're really missing the window of having this effective perturbation early. So what Xin need is a fast AAV that turn on as quick as possible. And very heroically, she worked with our collaborator, Xiangming Xu, and conducted this cross-phylogeny AAV screen. She combined together 86 different AAV vectors, and many of them were published in the past, comprising all these different serotypes and more. And what she did is to package this AAV with a green fluorescent protein, and each serotype was a unique barcode. So through this pool experiment, she can inject this AAV pool in utero at embryonic day 13 and wait it for 2 days. And after 2 days, if she see any green cells turn on, and that can allow her to harvest the green cells, sequence the barcode, and infer what serotype that was. So when I first learned that she did this experiment and waited for only 2 days, I was like, first-year graduate student. Usually you do an experiment for AAV, 5, 6, maybe a week. But I was very glad that Xin is right and I was wrong. This is what the brain looked like after 2 days of injection in vivo. And you can see that there are many green cells, both in the dorsal and ventral side of the developing brain. There are many different labeling across the lamin layer, which represents both the newborn neuron and neuroprogenitors. So we're very happy to have identified an AAV that can turn on as quick as 2 days. So accelerating gene delivery from weeks to 2 days. So this is just a different way to present the bulk sequencing data that of each of these rows represent AAV, and some of your favorite AAV that's been used here were highlighted. We do this analysis to compare the composition of the each of the AAV variants in the initial library, in vivo after 2 days, as well as in vitro experiment in the neuronal cell line. You can see that in vivo and vitro has quite different tropism or preferences. And on the right, this is just a different way to present the same data. And as you can see now, our top hit called AAV-SH9 is enriched in both in vivo and in vitro. And on the right is just a different way for you to see that although this vector was about 2.5% in the original pool, after 2 days, it really dominant in vivo pool in vivo from the GFP positive cells that comprise almost more than 25%. And this AAV-SH9 was first engineered from David Schaefer's lab. It was published a couple years ago. It really stood out as our top hits. So to fully characterize what this virus do, we perform the injection and have it to the brain postnatally 7 days after birth and perform the whole brain clearing and imaging. So what you're seeing is actually the signal collectively coming from the entire brain that is rendered transparent, and light sheet imaging going from the top to the bottom, as you can see from the right side of the movie. And if there's a brain region you are very interested in, you can go to this online neuro Lancet portal and zoom in to check your favorite region and the labeling density. But we were really excited to find out that it labeled nicely in our chosen sort of brain area of interest, that is the cortex, but it also allows us to access some brain region that's much harder to target, such as cerebellum, shown here on the right, the beautiful Purkinje cells here. And across the board, we found out that compared to lentiviruses, we can achieve modestly 4 to 20 folds of increase of density of labeling. That just means that you can do 20 times less work to get to the same amount of cell number. And there are some regions such as cerebellum that was never quite possible for lentivirus. Now AAV can reach. So integrating this AAV-SH9, the fast onset AAV system, and in conjunction with another tool that I'm not going to talk about today, which is a transposase system to further boost the transgene expression from AAV. Now Xin He conducted this in vivo Perturb-seq screen. She chose 4 key transcription factors, which heard plenty from Lacroix earlier, and these are a well-chosen transcription factor that was well studied in the past that are critical for corticogenesis, and we want to compare our results with the literature to see, as a benchmarking, what can we rediscover and what is the new discovery using the new higher resolution approaches. So I must emphasize that because we do this experiment in utero, in vivo, and after the injection, every day there's a new layer of neurons and or glia cells that's born. And what you're really asking is, upon perturbation, what are the cell type and different cell type contexts actually have different interpretation for these mutations. So throughout this analysis, Xin He collected over 2 big batches of experiment, over 50,000 cells, and each cell is labeled here in different color, indicating their really refined and distinct cellular properties in these UMAP plots. And I must emphasize that this is about 2 batches and reaching more cells that I personally could have in the 2020 paper, really emphasizing that the scale of the labeling, improved labeling, help us to in vivo experiments in the more sort of economic but also more robust ways. And in this time, using single-cell RNA sequencing, we actually did 5' sequencing, which allow us to actually reach to the target gene of interest and acquire the genotype of the CRISPR edits. We want to know whether CRISPR is really happening. And in fact, here are all the cells for different genetic perturbation. Each gene, we actually designed 3 distinct guide RNA there, as well as the control here on the right. So you can see that the cells were assigned to each this perturbation group. We can actually map and found out that for FOXG1, for example, the guide RNA 1 and guide RNA 2 groups, more than 60% of the cells actually have the transcript that has a frame shift insertion and deletion, whereas guide RNA 3 seems to have a weaker effect. And this is actually highly related to the phenotypic analysis that we observe. And it's also interesting to mention that there are variable guide RNA performances in vivo, which is what the entire field is facing, and it's important to consider them in analysis. This also gave us confidence because if we were not careful about the assignment of the perturbation, you might actually expect to see perturbation or these transcript captured in these other perturbation or control group, which is not the case here. And in the future, by leveraging new technology from mainly Dan Landau's lab, we can actually start to do this kind of genotyping as well as the phenotypic transcriptional analysis at a single cell level to start asking how the full homozygous knockout and heterozygous loss of function leads to potentially different phenotype in the future. So what have we learned from this screen? Here, I will briefly talk about the gene expression changes in distinct cell type across perturbation, one of the phenotypic analysis from Perturb-seq. So here, what we're looking at, each row represents a major cell type, and each column, again, represents a guide RNA belong to a perturbation group or the control. The dark shades indicates just simply the number of the genes. This is the 20,000-foot overview, and you can see that many of them, although variable across the 3 guide RNA, seems to have a concordant differential gene expression as a level of the disruption of the transcriptome. So if we zoom into this particular combo of excited neuron in layer 5, intertelephonic neurons, and upon the FOXG1 perturbation, we can go in and start looking at how different guide RNA leads to DE gene, and these DE genes are highly correlated. Again, as a validation for each other that we are actually truly capturing true changes in a particular cell type across different guide RNAs targeting the same gene. And I want to emphasize this other phenotype here that is in the layer 6 cortical thalamic neurons. For the aficionado audience, this is where the sensory input goes into the cortex, a very interesting cell type considering autism. And the loss of function of FOXG1 leads to really dramatic changes of differential gene expression agreed by multiple guide RNA. And here, well, we can see that there are many changes of the gene expression, both in terms of the adhesion molecule and axon guidance. And many of these genes were actually reported in the past 2 decades of traditional sort of knockout approaches that we can also observe in our approach, which is reassuring. But in addition, we also found out that there are many transcription factor labeled here all in red. And the loss of function of FOXG1 lead to an upregulation of these red molecules. The aficionado in the audience can notice that some of them are transcription factor. There are cell type markers or terminal differentiator almost to other cell type that does not belong to layer 6. And therefore, FOXG1's normal job is to repress these alternative transcription factor network. And after the removal knockout of FOXG1, these alternative cell fates start emerging. So just to summarize what I've told you in the progress of building these AAV-based in vivo Perturb-seq systems, we identify through a cross-phylogeny screen a fast AAV variant called SCH9 that accelerated gene delivery to 2 days. What I didn't tell you is the transposase that further improved the guide RNA capture in the single-cell analysis that led to the discovery of how these different transcription factor and their phenotype are extremely specific to the cellular context, such as the FOXG1 loss of function lead to the de-repression of transcription factor in the layer 6 cortical thalamic neuron that we do not observe in other excitatory neuron that's sitting the same layer or coming from the same developmental lineages, and underscoring the importance of using single cell and cell type specific approaches to start reading out these functional annotation of the genome. Last but not least, I want to thank our long-term collaborators, Joshua and Zhu Hao. My fundings, especially NHGRI, that was my first R01, which is really critical for the lab. And my lab is about 2.5 years old. We're actively recruiting. So if any of this is interesting to you, please send me an email, come talk to me. Thank you.
I was just wondering, so you had the 3 different guide RNAs and, one worked much better and one worked much worse in vivo.
Yeah.
How does that correspond to how they perform in vitro? Are they very similar or is it completely different?
Yeah, that's a great question. So I didn't mention that the 3 guide RNA that we ended up included in vivo were actually pre-selected in vitro. And then so far in our experience in general, they don't really correspond very well. But it does not mean that in vitro, we cannot do a little bit of optimization before going in vivo. Another approach that we and others started sort of employing is to put multiple guide RNA in single vector. So when you have one vector, you have multiple chances of inserting. Yeah, great question.
Very nice talk. I don't know much about AAV, but can you say something about the mechanism of why is it slow expressing and how the new variant fixes that mechanistically? Do you have an idea or no?
Yeah, no, that's a question we discussed intensely in the lab and also with many viral engineering lab like Bendevenham, Viviana, Guadagnaro. So we don't really have much of a mechanism, but we think it's just a more efficient virus. And we're in the process of doing sort of directed elution approaches to see if this AAV now is fast, maybe because its RT enzyme or copy number is higher, lead to higher expression. We can do capsid engineering probably to adding more modalities and hopefully not sacrificing this efficiency. But really, when you're talking about mechanism, we don't know how it works.